Thanks very much, Matthew, and to the organizers of the meeting. And again, really appreciate the colleagues from Israel being here. I, too, was hoping it would be in Israel. In fact, I had planned a trip to Europe, and I flew back late last night to get here as opposed to go to Israel late last night and for the meeting. But I'm very pleased to be here. So today, we've heard a lot about the Human Genome Project, and I'm pleased to say that this particular effort that you'll hear about, I think, is a combination of our understanding of the human genome and the cancer genome combined with AI and machine learning, and that, I think, will revolutionize many aspects of medicine in the years to come. And this is just a small example of how that may happen. So first, my disclosures, and then to say that this work for us really began almost 2 decades ago with some of the earliest cancer genomes. You can see here in a variety of different cancer types, initially performed actually with Sanger sequencing, given the dates that were here. A lot of new technologies were developed over time in this process. A number of new genes, and you heard about PIK3CA, and of course work that Yardena did, and other colleagues in the IDH gene discoveries, as well as other candidate genes, these leading to FDA-approved therapies. These have shown in many ways how complex cancer genomes are, revealed the complexity of this landscape, have laid the groundwork for the precision medicine that you just heard Arul talk about, identified that cancers change over time, and we need ways to understand that. And then finally revealed that perhaps a really important aspect which we need to think about is how to use this information for better diagnostic or avenues of detection of cancer. And I think detection is very important because cancer rate, despite all the work that we continue to do with precision therapy and immunotherapy and so forth, cancer rates continue to rise. You can see here by 2035, this number gets to be about 25 million, and the number of deaths is around 15 million. And there are a number of lines of evidence that suggest the largest potential strides against cancer will be in early detection. But for screening to work, we must find early stage disease, as this has the greatest impact on mortality. And screening also has to be a public health effort that's widely accessible at a population health scale, both in the US but also around the world. So a number of individual efforts have focused on using liquid biopsies or cell-free DNA for detecting cancer. And this began, of course, with the work of researchers in France over 50 years ago that found that there's free-floating DNA in the blood. And then several decades ago, researchers actually from the NIH discovered that DNA in the blood can actually come from cancer cells. And this led to the concept that looking just in a tube of blood, you can identify the particular characteristics of a cancer, detect it, and perhaps even understand how it changes over time. So a variety of methods began to be developed, and we were excited back in 2017 that Gillian Fallon led an effort to directly detect early-stage cancers using circulating tumor DNA. This was really the first systematic analysis of sequence alterations in cell-free DNA for direct detection of early-stage cancers. There had been a number of other studies, but many of them looked at the tumor first and then said, could we have identified the DNA in the blood? Now, although we were very excited initially when this was published, we quickly realized there's many limitations of looking at DNA. And we heard this morning about P53, the guardian of the genome, the most highly mutated gene in cancer, from work of Moshe and colleagues. But in this particular context, Alessandro Leal found that in a study of gastric cancer patients, the majority of mutations found in the blood were actually white blood cell derived. About 3/4 of these P53 mutations are coming from white blood cells. They're not in the tumor, they're just in the white blood cells, and only about 1/4 are tumor specific. And these are many of them at specific hotspots that you think of when you think about P53. So just a caution, when you're thinking of mutations and you're thinking about looking at the blood, changes in blood cells, in the more extreme cases, clonal hematopoiesis of indeterminate potential, are a major confounder for detecting cancer. So if this weren't enough, if you start thinking about cell-free DNA, there's a lot of cell-free DNA there. A lot of it may be coming actually from cancer patients, but if you think of the frequency of mutations, and you heard Arul speak about various ways to find mutations in cancer genomes, Well, they're probably about 1 in a million bases or so, sometimes more, sometimes less, depending on if you have mismatch repair deficiency. But that turns out to be the really tiny minority of all of the tumor-derived DNA that's there. So it made us think that we need to come up with better ways to detect tumor-derived DNA that's not based on mutations, given these weaknesses. And so one way that we developed was actually a next-generation liquid biopsy approach, as we call it, which is based on looking at low-coverage whole genome sequencing of cell-free DNA. This is work that was led by Stephen Cristiano. This methodology is called Delphi, stands for DNA Evaluation of Fragments for Early Interception. And this sequencing you'd expect to be uniform, that you're just sequencing the genome, you'd expect to get sort of equal representation. But it turns out that you get this fragmentation profile. The profile reflects the way the DNA is organized in the nucleus. The chromatin structure, areas of the genome that are open end up getting degraded, and are smaller areas that are closed end up being larger, and there's more of them. And you get this profile for healthy individuals. You get an altered one for individuals with cancer because the cancer cells are altered. You can ultimately use all these changes with artificial intelligence, machine learning, to identify those who have a cancer as compared to a healthy signal, and then further use machine learning to identify the tissue of origin because these signals contain an echo of the tissue from where they were derived. And so this work, and the work I'm going to describe to you, ultimately has led to a new ohm. We don't have enough ohms. We now have another one, the cell-free DNA fragmentome. The genome-wide compendium of cell-free DNA fragments in the circulation, providing an integrated view of the chromatin, genome, epigenome, and transcriptome states of normal and cancer cells. It's a lot of things there, and I'll describe some of the work that I think will hopefully persuade you that it does represent those aspects. So what does a fragmentome look like? Well, here's fragmentomes in healthy individuals. These are individuals without cancer. You can see there are 91 individuals here, and although there is a shape to them, they're very consistent. Here there's 67 individuals. These are individuals which have a benign lesion in the lung but no cancer. And then finally, individuals with lung cancer have these very widespread alterations in their fragmentation profile. These are the chromosome arms across the bottom, and this is the amount and size of DNA is on the vertical axis, a measure of that. And you can see within any one individual, there's many changes across the genome. Within the many individuals we're looking at, there can be many changes at any one of these chromosomes. Now, why are there fragmentome profiles to begin with? Why do we see something that's not consistent across the entire genome or equally represented, and there's a shape to it? Well, if you zoom in here on chromosome 1 and you compare the fragmentation profile to DNA that's isolated from blood cells, it's treated with nucleases, and you're essentially sequencing the lymphocytic nucleosomal DNA, you see a pattern that's very similar. So clearly this is, in a way, a representation of the chromatin of healthy white blood cells, and further evidence of that comes from high-C analyses. In individuals with cancer, of course, you have many things going on. You have genomic changes, chromosomal gains in red. You can see here in this tissue-based analysis of chromosomal arms in one patient. Gains are in red, the losses are in dark blue, and then the cell-free DNA is up here on top. You can see the same gains and losses, of course, in the fragmentation profile, but in addition to that, even in chromosomally stable regions, you see these changes in fragmentation profiles reflecting the chromatin aspects that are aberrant in cancer cells as well as other changes which we'll get to in a minute. And the benefit of this approach, which again is based on low coverage whole genome sequencing of cell-free DNA, very simple laboratory process, easy to do, inexpensive, is that in addition to this aspect of the fragmentation profile, you can obtain substantial amount of additional information. You can see here the chromosomal gains and losses we talked about, amplifications, deletions, rearrangements, which you just heard about many of in Arul's presentation. It's hard to get any single mutational signature, but you can get single mutation that you may be interested in, KRAS, etc. But you can get mutational signatures across the genome by looking at this low coverage data, transcription factor binding sites, mitochondrial representation, motifs, and then finally, I'll talk about it, the repeat landscape, the dark matter of the genome, which until recently was unavailable. With all of these different features and their genomic locations, working with colleagues like Rob Sharf, who we've been working very closely and collaboratively with for many years, we can essentially do machine learning, artificial intelligence to develop classifiers that are most suitable for detecting and identifying cancer compared to those which are present in healthy individuals. So we initially applied this approach systematically through different cancers to evaluate their utility for early cancer detection. And we started with lung cancer, and I already showed you the healthy individuals, those with benign lesions and those with lung cancers. Interestingly, it's important to be able to identify cancer from benign lesions. Many of the biomarkers from lung cancer actually can be confounded or fooled by that. This is not affected by that. And Dimitris Matthews led this study, which looked at over 300 individuals in collaboration with colleagues from University of Copenhagen. And he showed high performance, is an ROC curve, specificity, sensitivity for detecting lung cancer across all stages of lung cancer, across all the histologic subtypes, and even lung metastases. Now, this was very encouraging, but it was a smaller study, and it was from Northern Europe, homogeneous population, and also, although it was a prospective collection, it was retrospectively analyzed. So we have since then, in work led by Peter Mazzone, a renowned lung cancer expert, together with Peter Bach, one of the earliest proponents of lung cancer screening. Peter Mazzone's from Cleveland Clinic, and Peter was at Memorial Sloan Kettering prior to this work. And what this effort showed is that in a prospective study of over 1,000 individuals, about 50 sites nationwide in the US, essentially made to validate this approach, this effort showed a very similar pattern that Dimitris originally saw back in 2021. This is just published last year. So the individuals without cancer have very consistent profiles. The individuals with cancer have this very disparate profile. And this has now led, I'm pleased to say, to the first clinically approved liquid biopsy test for lung cancer screening that's ever been available. It is called First Look Lung. It's been launched by Delphi Diagnostics, which is a spin-out of Hopkins to take this technology to patients. And the test is intended for the 15 million individuals in the US who have or above age 50 and are at risk for lung cancer because of their smoking history. Typically, these individuals today are recommended for low-dose CT, but tragically, very few are getting screened. It's about 5 to 10%, and it's in large part because of the logistics of just getting a low-dose CT screening. So the idea here is to simply have a blood test that is easy to do, has high performance, and if positive would take you to the standard diagnostic workup, and if negative would take you to then annual screening. The key in such a screening test where you have an existing screening pathway is to have a very high negative predictive value. You need to be sure that the individuals who you call negative truly are negative, or else you might be sending people away from life-saving screening. So the negative predictive value of this approach is 99.8%. That's higher than the 99.5%, which is recommended for lung cancer screening. And this is just, obviously beginning to get off the ground. A number of health systems are using it, but estimates suggest that even a modest uptake in the United States would lead to thousands of lives being saved through prevention, essentially, through early identification and intervention in lung cancer. So we're optimistic and excited as this gets off the ground. There are a number of steps now in trials to take this beyond a clinically approved LDT test to an FDA-approved test and U.S. Preventive Services Task Force approval, and as well as trials in Europe that are going to lead to similar adoption in the European continent and hopefully around the world. So a lot more work to do, but early, I think, exciting days with this first lung cancer test. Now, the interesting thing about this is, as Eric was alluding to this morning, we've learned a lot about the human genome, we've learned a lot about the cancer genome, but there's a lot more that we can still learn, and we continue to realize that there's a lot of headroom here for detection of alterations in cell-free DNA that reflect what's happening in the cancer genome. And this is one example of that, work led by Daniel Broom, which essentially looked at genome-wide sequence alterations. And this was a very tough project. I said, Daniel, find out differences between patients with cancer and without cancer, looking at sequence changes in the blood at a coverage of essentially 1 to 2x. So single molecule sequencing. And what Daniel was able to show is that indeed, although you can't identify any one mutation with high, let's say, fidelity or accuracy, if you look across the genome, you find that there are mutational areas of increased mutational burden that are different in patients with cancer compared to those who are healthy. And there's well known that areas of the genome that are closed or not repaired as well, and they lead to higher frequency of alterations. And that's different, of course, in white blood cells compared to different tissues in the body. And so this different profile can be used with machine learning to identify healthy and cancer. This approach, called Gemini, works well on its own, but works even better when combined with the Delphi fragmentome approach, suggesting that these methodologies may be complementary. Moving on to other features like this, Ekshaya and Apragada decided to look at the repeat landscape. And we heard this morning a number of discussions about the telomere-to-telomere sequences, the long-read sequences you just heard from Arul. This has allowed the establishment now of reference genomes, which include a complete description of repeat sequences. Now, it's only in a small number of individuals. More will be underway. And you actually can't use long-read sequencing in cell-free DNA because the DNA is fragmented. It's very short. It's only about 170 bases. So what Ekshaya devised was a way to classify, now that the telomere-to-telomere sequence was established, about 1,000 repeat element types can be quantified using short-read sequences to identify their level in cell-free DNA. This doesn't tell you where in the genome they're coming from, but it allows you to look at these classes and identify whether there's a change happening in individuals with cancer versus healthy. She showed this in cancer tissues, showed there's widespread differences, probably the first time such a systematic analysis of repeat elements in human cancer has been done, and then she went on to show it in cell-free DNA and showed that alone it too has pretty high performance, but again, combined with fragmentomics, it's even higher. This is in the same lung cancer cohort. And these types of features keep continuing. So here's an example of looking at fragmentation changes or sequence coverage at transcription factor binding sites. In this case, it's at the binding sites of pioneer factors that are frequently expressed in small cell lung cancer. This is ASCL1. And in looking at the binding site of these 13,000 transcription factor binding sites, you can easily distinguish small cell lung cancer from non-small cell lung cancer. Probably the first time a liquid biopsy has distinguished between these two lung cancer subtypes. This is work led by Dimitris Matthews in the initial lung cancer study. And with high ROCs, we can now even not only detect lung cancer, but potentially distinguish the different subclasses. More recently, Michael Noe showed that methylation actually affects cell-free DNA fragmentation. It looks like CPGs increase in the methylation at CPG sites leads to a higher fraction of fragments having an end position. The cell-free DNA has a start and an end, and that end position can be at CPG islands, and that is increased when it's methylated. It's decreased when it's a CCG because of the competing CPG here. And the reason for this appears to be because of the structure of cell-free DNA. Essentially, it's a wrapping of DNA around a nucleosome, and the presence of CPGs at these sites here, close to this linker histone, ends up stabilizing the cell-free DNA, making it harder to degrade, while if there's no methylation, the DNA flops open and it can be degraded and ends up being smaller. And then in addition to the end positions, the amount of cell-free DNA is higher when there's methylation, lower when there's no methylation, and similarly, low gene expression, genes that are not expressed have higher coverage, and high expression ends up leading to lower coverage. So you can use this kind of information, again, to detect, in this case it was pancreatic cancer, cancer that's hard to detect because of the collagen you heard about earlier, makes it hard for the DNA to come out, but it could be detected using these approaches, and together they can be combined. So essentially, all the different characteristics that you think about as being aberrant in cancer, starting on the left with genome instability, abnormal chromatin, altered nucleosome occupancy, altered gene expression, genomic and epigenomic changes to repeat landscapes, and finally methylation changes. Of course, these are altered in cancer compared to healthy individuals, and essentially all of them can have an impact on the cell-free DNA. And these potentially can be measured, and we've been working sort of systematically over the years to develop approaches that can identify these kinds of changes, starting actually more than 15 years ago to identify chromosomal changes using digital karyotyping. Then we've talked, and this is in cell-free DNA, and then thinking about fragmentation, we talked about the Delphi approach both globally and locally and let's say focally. Mutational signatures, the Gemini approach, repeat landscapes, and then finally epigenetic changes. And I think there'll continue to be such alterations and kinds of changes that we can now uncover in cell-free DNA. And so in the very last few minutes, I wanted to just cover a few additional applications that we've been pursuing in other cancer types using this technology, not currently employing all the different features we talked about, but really the initial approaches as we described them. And the first was in liver cancer, and this is a disease that's terrible. It's the third leading cause of cancer death worldwide. There's a population at risk of about 400 million individuals, typically hepatitis and cirrhosis. And so Zach Foda and Akshana Pragada led an effort to see if we can identify in about 700 individuals those who have cancer versus those that don't. This is hepatocellular carcinoma fragmentation profile, those without cancers, then finally those with hepatitis and cirrhosis. No changes here, perhaps a few changes here, and a lot of changes obviously in HCC. Very high performance for detecting this cancer type using this approach across the different Barcelona Clinic liver cancer stages, those that are resectable 0A and a part of B. High performance overall in a cross-validated cohort, and high performance using this locked approach in a validation cohort from Hong Kong. There's additional validation cohorts which Zach Foda is pursuing, and it looks very encouraging. And this, I think, will be something that'll be very useful worldwide. The second deadly cancer which we focused on has been ovarian cancer. Ovarian cancer is a leading cause of death in women worldwide, with more than 300,000 new cases and 200,000 deaths globally each year. And unfortunately, there's no good biomarker. The one biomarker that's been evaluated on its own, CA125, has failed. So we evaluated whether fragmentation profiles together with CA125 and HE4 can make a better combined protein biomarker, work led by Jamie Medina and Akshana Pragada. High performance across the different stages using this combined approach, and the locked model here, including in high-grade serous, which is the one most responsible for deaths in ovarian cancer, and the locked model here evaluated in a validation cohort. Smaller numbers, but encouraging data with high performance so far. And I think the most encouraging part is that the performance ranged from 70% in stage 1 to 100% for this Delphi Pro approach, while C125 in stage 1, the most important, let's say, place to detect, is only about half that. And in this particular setting, one actually needs a very high, what's called positive predictive value. You need to know that the individuals who are predicted as positive, a high fraction of them will have cancer, because the next step, there is no diagnostic pathway. The next step is really, in many cases, a surgical laparotomy. And so the recommended PPV here is above 10%. This approach is about 18%. This is about 6%. And so that shows why perhaps this failed. And there's possibilities, I think, for this. So there's exciting, I think, follow-on work to do here. And then finally, I wanted to share with you a story of detection of brain cancer. Brain cancer has essentially been undetectable in the circulation. If you look for mutations, you only detect it about 5% of the time. This is even for GBM, and it's presumably because of the blood-brain barrier. And so Dimitris Mathios, a neurosurgeon who we recruited to do the lung cancer effort initially, and Nusha Niknafs, pursued this study initially in a discovery cohort, and then following up. This is over 500 individuals with or without brain cancer, initially in a discovery cohort, and then in a validation cohort using the locked model and evaluating the performance in this setting. And surprisingly, this approach detected brain cancers at a higher, with a higher performance. This is individuals with cancer across different grades. GBMs are here on the right, versus those without cancer or non-cancer neurological controls. This model was then evaluated in a validation cohort, still had relatively high performance, and you can see here the ultimate performance is, in the, let's say, 70% range across these different grades, much better than was done before. And so one of the questions, well, what's the underlying biology? Why is this possible? And it turns out that if you look at the fragmentome, let's say, surrogate of the transcriptional changes that are going on in cell-free DNA, you're getting signal that's coming from the brain, astrocytic signal here. You expect a reverse correlation here, negative correlation, but you're also getting signals from white blood cells. And this matches the known characteristics of brain cancer, which end up leading to essentially widespread immune suppression in patients with GBM or even earlier grade gliomas, suggesting that this approach is capturing fragmentomic changes not only coming from the cancer and the DNA coming from the cancer, but DNA coming from changes in white blood cells. And with that, let me just say that we're, of course, in the long run, interested in systematically going through individual cancers where there's a diagnostic pathway, but ultimately going to multi-cancer. The initial study in Nature described this in a multi-cancer setting, 7 different cancers, and even showed possibility of identifying a tissue of origin. And let me just conclude by saying that, I think today we've talked about using machine learning and this fragmentome approach for detecting cancer, including in early stage disease. The approach in lung cancer has now been validated for clinical use, and I think that that's an encouraging first step in that disease. And the approach shows promise for early detection of a number of other cancer types. We've talked about liver, ovarian, brain. There are others that are underway. And it may be useful also for detecting response or resistance to cancer therapies. Didn't talk about this at all, but this can measure responses over time. Then finally, I think these kinds of accessible analyses can provide a path for early detection, could be implemented worldwide. Just wanted to recognize all the folks that made this work possible in our group at Hopkins, other institutions that we work closely with, the investigators, and most importantly, the patients participating in the prospective clinical trial, and finally, members of our group and our funders. And with that, I want to thank you and happy to take any clarifying questions.